Hello, Mr. P here. Welcome to the last lecture of Chapter 10. This is the fourth lecture, and in this lecture we're going to talk specifically about cell cycle regulation. We've already talked about what mitosis and what cell division really is, and we've talked about the phases of each, specifically G1, S, and G2 of interphase, and PMAT, or prophase, metaphase, anaphase, telophase of mitosis. Remember, mitosis is the division of the nucleus, and interphase is the division interphase is the cell's life cycle prior to cell division. But let's go ahead and talk a little bit about cell cycle regulation. It's important to note that the cell cycle, which we've talked about before, is regulated, and we're going to see later on, or a little bit later on, that there are things that are going to control the cell from inside the cell, and there are things that are going to control the cell from outside the cell. These are called internal and external regulators, but for now, we're going to kind of talk about why the cell needs to be regulated. And it's really important to note that controlled cell growth and controlled cell division is vital to the proper cell function, meaning a cell isn't going to be able to do the things that it needs to do unless it's controlled. That's one of the reasons why the cell, or eukaryotic cells, have a nucleus. It's one of the reasons why cells have DNA. And just like you, it's a lot easier and a lot better for you to live a very controlled life rather than live a very wild, uncontrolled life. And cells are no different. And so cells wear out. We've talked about the whole purpose of cell division is to fix worn out cells that have been broken down or have been injured, or from an organism standpoint, mitosis and cell division is vital in order to fix wounds. I think everybody's probably had a sunburn, or somebody's probably fallen down and had a scrape, scraped your knee or scraped your elbow, or somebody's probably needed stitches, somebody probably has broken a bone. All of these things happen to you as an organism, and it's vital for you, basically, it's vital for your wound healing processes that cells are controlled and that cells will fix each other, okay, and fix themselves. So cell division is really usually pretty slow, and cell division increases when the cell is damaged or the cell is removed. And so we're going to talk a little bit more about that as we go. But one thing that's really important to note is that cell division requires cyclin, and cyclin proteins are get a pin here. Cyclin proteins are vital to this whole regulation pathway, and so we're going to talk a lot about cyclin. And it's cyclin, not cycling. This is all wrong in so many ways. But while we're talking about cyclin, or specifically cycline, I got a joke for you. Why did the bike fall over? It was too tired. Alright, sorry about that. He got a little carried away. Anyway, we need to talk about cell cycle regulation, and you have a vocab word, and one of the vocab words is cyclin. Okay, cyclin is a specific protein that regulates the cell cycle in eukaryotic cells, and cyclin alone can't really do anything. It requires a cyclin-dependent kinase, which is an enzyme that it's going to bind to in order to regulate the cell cycle at specific points in that cell cycle. Okay, and I'm going to get into it a little bit later where each of the CDK molecules and where the specific cyclins are going to work. But for now, remember back in chapter 2, I believe, when we talked about enzymes, there is an enzyme. This is the enzyme. Okay, in this case, the CDK. There is an active site and there is a substrate, and these molecules will bind to each other, and the cyclin's job is kind of to turn on the cyclin-dependent kinase. Okay, so these CDKs are going to be turned on or activated by the cyclin proteins, and as soon as you have kind of the cyclin-CDK complex, then it's going to allow the internal and external regulation of the cell cycle. Okay, and so where is cyclin used? notice that there is cyclin A, there is cyclin B, cyclin D, and cyclin E. And there are phases down here, G1, S, G2, M. And so as the cell cycle progresses this way, from G1 to S to G2 to M, you will see that there is a peak concentration of each of these cyclin proteins. Okay, so in G1, when the cell is growing and when the organelles are replicating, there is a fairly high amount of cyclin D. And as the cell cycle progresses to S phase, when it needs to replicate the DNA, there is a huge influx or a huge concentration, increase in concentration of cyclin E. And as you go towards G2, which is the last phase of interphase, there's a big peak in cyclin A. And as you go to M phase, which is the actual cell division, or the mitosis is the division of the nucleus, there's a huge peak of cyclin B. Okay, so DEAB, it's like DAB, like DAB. I mean, I know you all know what DAB is, but if you want an acronym that might help you learn the order of these cyclin proteins and the order in which they're used, I've got a couple acronyms for you. DEAB. Does every aardvark like blankies? You betcha. That's an ugly little critter. Dancing elephants are banned. Absolutely. That elephant's definitely breaking it down, but no. Regulating the cell cycle. Cyclin and CDKs. Remember, the cyclin is a protein that is needing to bind to a CDK. So cyclin is a protein, CDK is the enzyme. And if you look at the cell cycle, G1 is the first phase of interphase, S is the second phase of interphase, G2 is the third phase of interphase, and then M is mitosis. Okay, and the cytokinesis would be right here. So we're kind of starting and stopping here. And I'll put a little star here. Start and stop. And so you'll see where these specific CDK proteins or CDK enzymes and where the actual cyclins are going to be regulating the cell cycle. Okay, cyclin D, E, A, B, right? Dab. It's like if cowboys, if it was popular to dab in the cowboy days, they'd be like, I'll be dabbing all day. Dab. I don't know what they're doing right now, Dab. Anyway, moving on. Internal and external regulation. Okay, this is going to be important for you to know. Internal and external regulators. Internal is exactly what it sounds like. They're going to be regulating the cell cycle from inside the cell, and these specific proteins are going to be responding to events that happen inside the cell. External regulators are proteins that respond to events outside the cell. Okay, and so we're going to talk about the specifics of internal and external regulators. Internal regulators are going to allow the cell cycle to proceed, okay, allow the cell cycle to proceed when certain interior processes have happened. There are these things called regulatory proteins. These regulatory proteins are going to, in the first example, and these are both examples, going to prevent the cell from entering mitosis until all the chromosomes have been replicated. Where do the chromosomes replicate themselves? They're replicated in S phase. Okay, and so there is a checkpoint. There are regulatory proteins that are going to check for whether the chromosomes have replicated not only fully but accurately. Okay, and if they don't, if they haven't replicated accurately or completely, then these regulatory proteins are going to stop the cell cycle from progressing any further. Because why does the cell, or why would the cell want to progress if all of the DNA hasn't been replicated? That's just setting you up for failure, or setting the cell up for trouble. Okay, we don't want the cell in trubs. The cell doesn't want to be in trubs, and so there are regulatory proteins that are going to prevent the cell from entering the next phase if everything prior to that phase hasn't happened yet. And so this is one example. The second example would be regulatory proteins preventing a cell from entering anaphase. And remember, anaphase is the third phase of mitosis until all the chromosomes are attached to the mitotic spindles. Why would that be important? What happens in anaphase? Well, hopefully you said, and I can't hear you, but hopefully you said that the chromatids are separated in the anaphase. Okay, and so if the chromosomes haven't been completely attached to the mitotic spindles yet, then you don't want to progress to anaphase, because if anaphase progresses and starts pulling the chromatids apart, but not all of the chromatids have attached to the spindles, then you're not going to get an accurate separation of the chromosomes, and you're going to end up with more chromosomes in one cell versus the other, and that could lead to all kinds of problems when we talk about mitosis wanting to produce identical daughter cells. Okay, and so when we talk about external regulation, these are proteins, again, that are going to respond to events that are happening outside the cell, and this is more noticeable, or I guess more involved with direct cell-to-cell communication and the speed in which the cell is dividing. And so the cell will speed up or slow down depending on how much contact it has with neighboring or adjacent cells. If the cell, and I'll just draw a little example here, if the cell is all by itself, it's going to speed up because externally speaking, it's not communicating or touching other cells. Okay, let's just say there's a cell here and there's a cell here. There's all this space in between. Well, this cell is going to become 2 cells, and those cells are going to become 4 cells, and those 4 are going to become 8, and eventually they're going to keep replicating, and you'll end up with a bunch of cells. Well, when this cell eventually replicates and touches this cell, these are now communicating, and because they are kind of encroaching now on each other's space, they're going to start slowing down, because we don't want to kind of choke out each other, cellular speaking, right? We don't want to make a mass of cells. We want to make a nice even layer of cells, and that's what tissues are, right? So when we're talking about skin, your skin is nice and smooth, and it's not jaggedy, and it's not bumpy, and your skin isn't varying thicknesses. Your skin is pretty much of one thickness, and it's this reason, these growth factors, that are going to do that, and we'll talk a little bit more about what that is. But internal regulators, remember, are proteins that respond to events that happen inside the cell. External regulators are proteins that respond to the events outside the cell. So what are growth factors? Growth factors are going to be those things that are external regulators. Okay, they're happening outside the cell or responding to signals that are coming from outside the cell. And proximity is really important. Proximity means how close the cells are to other cells, and cell size is another one. And chemicals can and will dictate when the cells divide as well. So imagine these 2 cells growing, and all cells do grow, but if this one gets a little bigger and a little bigger, and all of a sudden it touches this cell, well, this growth factor is going to basically talk to the cell, externally speaking, and it's going to stop the growth of this one because it has already touched the neighboring and adjacent cell. It doesn't need to grow anymore. It's already filled the space that it was allotted, and so that's what these growth factors are for. Growth factors attach to receptor proteins in the cell membrane to trigger cell division, and cells generally stop growing and maintain themselves through G1. Remember, cell growth occurs during gap 1, and gap 1 and interphase really is the kind of the maintenance or the life cycle, if you will, of the cell. That's where the cell's doing all of its cellular games. If the correct conditions exist, growth factors present, cell will continue through the cell cycle and eventually reach mitosis and eventually divide into 2 daughter cells. Okay, so here are the checkpoints. This is G1. Okay, so there's a G1 checkpoint. Then you go to S, where the DNA is replicated. Then you go to G2, where the additional growth, the production of enzymes, and the additional replication of organelles happens. And then there's a G2 checkpoint. This checkpoint will dictate whether or not the cell will enter M phase. Once it leaves checkpoint 2, or the G2 checkpoint, it will enter mitosis, which is prophase, metaphase, anaphase, telophase. Again, remember, we said that cyclin, that there is a cyclin CDK combination that happens during S phase, and it's going to specifically check for whether or not the mitotic spindles have attached to the kinetochores of a particular chromosome, and we want to make sure that all the chromosomes at this point have connected to that spindle apparatus, so that when anaphase happens and all of the sister chromatids are separated, we can ensure that all sister chromatids have separated, and each new cell will have a full set of chromosomes after that. Okay, G1 checkpoint, G2 checkpoint, M checkpoint. All three are important. However, G1 is kind of considered the most important step because the vast majority of cells that go through checkpoint 1, the G1 checkpoint, if they get past this, chances are, even if they mutate in this step, or in case something happens here, chances are they're going to enter M phase. This is the most important checkpoint for the cell. This is kind of the make it or break it. These are kind of backup, but these aren't as efficient, and so they're not as good as G1 at correcting errors or identifying errors. G1 checkpoint is the main checkpoint. So how do growth factors work? Growth factors are, remember, external regulators, and so these are signals that are going to be sent to a receptor protein that are embedded in the plasma membrane. Remember in Chapter 7 when we talked about membranes, we did a lot of protein discussion, okay, or we had a lot of protein discussion, and some of the proteins, remember, are globular proteins. Some of them are more structural proteins, but there are a lot of different proteins that do a lot of different jobs, and one of the proteins that are embedded in the membrane are receptor proteins, and these receptor proteins are going to receive a signal in the form of a growth factor. They're going to basically relay that information or that signal to the first of many relay proteins, and these relay proteins are going to take that signal to the kind of the theoretical control system of the cell cycle. Now, there isn't a little spinner like there is on board games within the cell that's kind of regulating where the cell is at any given moment, but you get the idea. These relay proteins are going to relay this message that is coming from the outside of a cell to the control center, the thing that is controlling the cell cycle. And if they proceed, or if they perceive them as being ready to exit G1 into S, then they will pass that checkpoint and will eventually progress through the cell cycle. If, however, the growth factor tells the cell to stop dividing, like it has reached other cells, or it doesn't have any more room to grow, then it's going to basically put up that checkpoint wall, and it's going to block the progression of that cell cycle throughout the cell cycle. So, what happens when cell cycle regulation errs? Okay, or what happens when the cell becomes unregulated? Well, you get cancer. If you were to put normal cells into a petri dish, they would grow in a nice, uniform, very I don't know what the word is, very uniform layer of cells. However, if you were to mutate one of the cells in the middle of this nice uniform layer into a cancerous cell, and that cell becomes unregulated, then it's going to increase the speed at which it divides, and you're going to get a mass of cells. Notice they don't grow in a nice even layer like the non-cancerous cells do. They grow in a mass. They're going to grow up, and they're going to grow down, and they're going to grow out to the sides, and they're going to grow up at diagonals, and they're going to go down at diagonals. They're going to grow in every direction from that center cell. They're not regulated anymore. And if they form this lump in normal tissue, it's what we call a tumor. Okay, and I know probably everybody has talked or heard about tumor. Tumor is an abnormal mass of rapidly dividing cells. Why are they rapidly dividing? Because they are no longer controlled. All of that CDK, cyclin combinations, all of the growth factors, all of the checkpoints, all of that stuff that normally controls cells has been turned off of these specific oncogenes, and the genes have been turned on that kind of cause the cell to become unregulated, and we basically turn everything we want on and everything we don't want off, and you get a mass of cancer cells that we call a tumor. So how does cancer spread through the body? We know it as metastasis, okay? And so cancer cells can mutate randomly, and there are things called carcinogens that will increase the chance or increase the likeliness of a specific cell becoming cancerous or becoming unregulated. And so if this one cell becomes unregulated, then it divides into two, maybe this one and this one, and then those divide into maybe this one and this one, and then those 4, maybe this one, this one, this one, this one, and you get the idea. But it grows every which way into a tumor, into a mass, and if they grow into a blood vessel, notice that normal cells, due to that proximity and growth factors, normal cells will actually stop dividing when they reach another tissue type, or when they reach a blood vessel, or when they reach a bone, or when they reach a muscle. But cancer cells don't stop, and so they will continue to grow and grow and grow and push on these blood vessels and push on other tissues, and eventually some of these cancerous cells can actually push their way into the blood vessel. And once cancer cells that are unregulated and become basically rapidly dividing, once they get into the bloodstream, they can be deposited other areas in other tissues and cause a new tumor there. Okay, and so that's called metastasis. There are differences between tumors, okay, or among tumors. There are benign tumors and there are malignant tumors. Benign tumors are the good tumors, and I don't necessarily want to use good and tumor at the same time because there are no good tumors. But the benign tumors are the ones that are easy to deal with. They're the ones that can generally be removed. They're the ones we consider non-cancerous. Okay, and so I guess if you're going to get a tumor, if you're going to know somebody that gets a tumor, you want it to be benign. Benign tumors can be kind of fatty tissue in nature. They can be other tissues in nature, but they're generally easy to be removed. And malignant tumors are the one that spread, whereas benign tumors are generally not spreading. They stay in place, they stay put. And malignant tumors are the bad or the worst tumors. They're the ones that spread to neighboring tissues. They're the ones that actually cause cancer. They are the worst to deal with, and these are the tumors that people need chemotherapy or radiation therapy for, because these are ones that are not going to be easily dealt with. Benign tumors can be easily dealt with surgery. So I've talked about cell cycle regulation, and in the last 3, or the previous 3 lectures, we've talked about mitosis and cell division and somatic cells and gametes. And so at this point we need to stop talking about mitosis because it's such a divisive issue. But this is the last lecture of 2016. When you guys return after Christmas break, we will continue our journey through this biological world. Until then, see ya.